Hello students, today's topic of discussion is digestion and absorption of proteins. This is an important topic and can be asked as SAQ, short answer question, in university examination. So first, keep the thing in mind that whenever there is digestion of any compound, the meaning is the conversion of these macromolecules, means the bigger molecules, into a smaller unit. For example, digestion of carbohydrate. If digestion of carbohydrate is there, the digestion means breaking this carbohydrate, means breaking the starch or glycogen or polysaccharide unit into its constituent monosaccharide, means the glucose or monosaccharide. Okay. Whenever it is digestion of lipid, it means conversion of these lipids into fatty acid and glycerol. Similarly, whenever there is digestion of protein, it means these proteins are broken down into their constituent amino acids, and then these amino acids, they are taken up by the intestinal mucosal cell and absorbed, and it is in the circulation, that is known as absorption. So this is known as digestion, and when it is taken up by the intestinal mucosal cell, it is known as absorption. Through the mucosal cell, it is into circulation, that is known as absorption. So digestion and absorption of these macromolecules, means protein, carbohydrate, lipid, these are important, and these are important SAQ, short answer questions for university examination. Okay, coming to the digestion of protein. In digestion of protein, the proteins, they are actually they are made up of amino acid. How will you define protein? These are the polymers of L-alpha amino acid. So in the digestion of proteins, proteins are converted into amino acids. So coming to the discussion of digestion first, then we'll discuss the absorption. In digestion of proteins, these proteins are broken down into constituent amino acid. So three important organs, they play important role. For example, stomach, the first one is the stomach. It is important. Then intestine and pancreas. Intestine and pancreas. These are the important organs which take part in the digestion of proteins. There is negligible digestion or no digestion of protein in the mouth. Then in the stomach, there is hydrochloric acid. Gastric juice contain hydrochloric acid. Two important role of hydrochloric acid. First, it causes the denaturation of the protein. What is the meaning of denaturation? Denaturation means loss of secondary, tertiary and quaternary structure, and only the primary structure remain intact. So suppose this is tertiary structure of the protein, and in the denaturation, this tertiary structure is broken down into a primary polypeptide. So internal peptide linkages. If there are internal peptide linkages, now these are free for the action of the peptidases. These peptide linkages are exposed for the action of peptidases. The peptidases means the enzymes which act on the peptide. So peptidases. So this is the action of HCl, that is the denaturation. Second important role of HCl is there is secretion of pepsinogen, which is converted into pepsin by the action of hydrochloric acid. Pepsinogen is activated into pepsin by the proteolysis by cutting the small peptide. Suppose this is a pepsinogen. By cutting small peptide, by removing small peptide, it becomes active. That is known as pepsin. It is known as pepsin, and it is active in the acidic pH of the stomach. So pepsinogen is converted into pepsin, and pepsin is the endopeptidase. This is an endopeptidase. What is the meaning of endopeptidase? These enzymes act on internal peptide linkage. These are the internal peptide linkages. This is known as endopeptidase. And exopeptidase will act on the terminal peptide bonds. Suppose this is amino acid, the exopeptidase will cleave these bonds. Exopeptidase act on the terminal, either the N-terminal or carboxyl terminal amino acid, or N-terminal peptide bond or carboxyl terminal peptide bond. This is the action of exopeptidase, while this is endopeptidase, act on internal peptide bond. So pepsinogen is converted into pepsin, and third important is the renin. Renin. It is chymosin. It is an enzyme present in the infant. It is not present in the adult. It is present in the infant. Renin causes the curdling of the milk. Renin act on the milk casein. It act on milk casein and converts this into calcium paracaseinate. Convert the casein into calcium paracaseinate. This is a solid insoluble form. Insoluble form, and the time it takes more time in the stomach. So the action, but then it is acted upon by the pepsin. So milk, for the milk, the transit time in stomach is very less. It is cleared up easily. Milk is cleared up easily. So renin act on the milk, causes the curdling of the milk, so that converts the milk into solid insoluble form, and it is the time, it gives the time for the pepsin to act on this calcium paracaseinate. So this is important here. It is present in the infant but not present in the adult. Renin. Okay. So HCl, pepsinogen and renin, it is secreted in the stomach. Now when food bolus reaches the intestine, there is action of CBSE. There is action of CBSE. Remember this, in the intestine, there is release of cholecystokinin from the duodenum. Specifically the eye cells of the duodenum, there is secretion of cholecystokinin when food reaches the duodenum, along with the secretin. And this secretin causes the release of bicarbonate from the pancreas and neutralizes the acidic pH of the stomach. For the pancreatic enzyme to act, pH required is near about 8. So secretin causes the release of bicarbonate. Secretin, it is secreted by the S cells of the duodenum and jejunum. So secretin causes the release of bicarbonate from the pancreas. And enteropeptidase or enterokinase, this is released from the mucosal cell of the intestine, and it particularly activates the trypsinogen. It activates the trypsinogen, convert trypsinogen into trypsin. So role of these enzymes, it is very, very important. Cholecystokinin, bicarbonate, secretin and enteropeptidase are enterokinase. These are the important enzymes secreted by the intestinal cells. Okay, now the pancreas, this is the important one. The pancreas, there is release of, from the pancreas, there is release of peptidases, particularly the endopeptidases and some exopeptidases. They are released from the in the pancreatic juice. Okay, so enteropeptidase causes release causes the conversion of trypsinogen into trypsin. Cholecystokinin causes the release of pancreatic enzymes, peptidases. So there are trypsinogen, chymotrypsinogen, pancreatic peptidases. These are trypsinogen, chymotrypsinogen, proelastase, and procarboxypeptidase. Trypsinogen, chymotrypsinogen, proelastase, procarboxypeptidase. They are secreted as inactive zymogens, inactive enzymes. And by the proteolysis, by the limited proteolysis, these trypsinogen or these latent enzymes, they are converted into their active form. So first enteropeptidase causes the conversion of trypsinogen into trypsin. Then trypsin causes the conversion of chymotrypsinogen into chymotrypsin, proelastase into elastase, procarboxypeptidase into carboxypeptidase. And then there is role of mucosal cell. From the mucosal cell, there is release of aminopeptidase and dipeptidase that will act on the smaller peptide and converts the smaller peptides into a free amino acids which get absorbed from the intestinal mucosal cell. Okay, now see. So protein, what do we eat? Protein or polypeptide. It is in the stomach acted upon by the HCl. So HCl denaturates the protein and causes the conversion of pepsinogen into pepsin. So pepsinogen is converted into pepsin. Now what is action of pepsin? Pepsinogen is converted into pepsin. And what is action of pepsin? It cleaves the internal peptide linkage. Suppose this is polypeptide. This is carboxyl terminal. This is amino terminal. So pepsin will act on internal peptide linkages. And pepsin specifically act on the N-terminal phenylalanine, tyrosine, tryptophan, leucine, and some acidic amino acid. It acts on the peptide bond formed by the N-terminal. This is N-terminal amino acid. This is N-terminal. It acts on the peptide bond formed by the N-terminal amino acid, N-terminal phenylalanine or tyrosine or tryptophan or leucine or some acidic amino acid. On that peptide linkage, pepsin will act. It is active at the acidic pH, pepsin. Acidic pH which is present in the stomach and activated by the HCl. Also autocatalysis means pepsin itself activates the conversion of or causes the conversion of pepsinogen into pepsin. Got it? So this is in the stomach. So proteins, they are converted into, by the action of pepsin, proteins, they are converted into peptides or oligopeptides. Then it is acted upon by the pancreatic enzymes which are released in response to cholecystokinin. When food reaches the duodenum, cholecystokinin causes the release of pancreatic juice and secretin causes the release of bicarbonate which provides the pH. Optimum pH for pancreatic enzyme is 8. Now this peptide, oligopeptide, they are acted upon by the trypsin, chymotrypsin, elastase and carboxypeptidase A and B. Now see, this trypsin, it will act on the carboxyl terminal. It acts on the peptide bond formed by the carboxyl terminal arginine and lysine. Peptide bond formed by the carboxyl terminal arginine and lysine. Chymotrypsin will act on the carboxyl terminal aromatic amino acids, that is phenylalanine, tyrosine, tryptophan, then histidine, aspartate, methionine, leucine. So it will act on the peptide bond formed by the carboxyl terminal phenylalanine, tyrosine, tryptophan, histidine, aspartate, methionine, and leucine. So see this. This is the N terminal. If this is the action. So now this chymotrypsin will act on the carboxyl terminal. Suppose this is carboxyl terminal phenylalanine. So it is acted upon by the chymotrypsin. Okay, and the trypsin specifically act on the peptides which escaped digestion from the pepsin. Okay, so this is the mechanism of action. Elastase will act on the carboxyl terminal glycine, serine, alanine, and carboxypeptidase will act on this carboxyl terminal exopeptidase. This is exopeptidase, so it will act on this carboxyl terminal amino acid, and there is formation of smaller peptides, dipeptides, amino acid, free amino acid liberation. Carboxypeptidase will leave the free amino acid, will remove the free amino acid. So there is formation of peptides, dipeptides, and amino acid. These are further acted upon by the dipeptidase, then aminopeptidase from the intestinal mucosal cell, and there is formation of free amino acids, dipeptidase and aminopeptidase from the intestinal mucosal cell. How the activation of enzymes occurs? See, the enterokinase or enteropeptidase or enterokinase, this causes the conversion of trypsinogen into trypsin. Now trypsin catalyzes the conversion of chymotrypsinogen into chymotrypsin. It also catalyzes the conversion of proelastase into elastase. Procarboxypeptidase into carboxypeptidase. So this is by the trypsin. Trypsin causes the conversion of chymotrypsinogen into chymotrypsin, proelastase into elastase, procarboxypeptidase into carboxypeptidase. Once again, we will revise the mechanism of action. So pepsin will act on the amino terminal or N terminal, will act on the peptide bond formed by the N terminal phenylalanine, tyrosine, tryptophan, leucine, and some acidic amino acid. Okay, the peptide bond formed by the N-terminal phenylalanine, tyrosine, tryptophan, and leucine. The trypsin specifically act on the peptide which escape digestion from the pepsin. It particularly act on the carboxyl terminal arginine and lysine. Chymotrypsin, it will act on the carboxyl terminal phenylalanine, tyrosine, tryptophan. These are the aromatic amino acids. Then it will act on the methionine, leucine. It will act on the aspartic acid, histidine. The carboxyl terminal amino acid. The peptide bonds formed by the carboxyl terminal amino acid. Elastase will act on the carboxyl terminal glycine, serine, alanine. Glycine, serine, alanine. The carboxypeptidase will act on the carboxyl terminal amino acid and will free the amino acid. Suppose this is peptide, this is carboxyl terminal, this is amino terminal or N terminal, that is also known as amino terminal, and these are the amino acids. So this on this end, this is exopeptidase, carboxypeptidase, this is exopeptidase, carboxypeptidase A and B. This will cleave this amino acid and will remove this amino acid. While aminopeptidase will act on this peptide linkage and will free this amino acid. These are the exopeptidase, carboxypeptidase, aminopeptidase, these are the exopeptidase. While rest are pepsin, trypsin, chymotrypsin, elastase. These are the endopeptidase. These endopeptidase specifically act on internal peptide linkages. Internal peptide linkages. So they will act on the peptide bonds formed by this particular amino acid, either N-terminal or C-terminal. So these are the different enzymes which act on the protein or peptide and converts the larger polypeptide into smaller fragments. Now these smaller fragments further acted upon by these peptidases and they are converted into oligopeptide, dipeptide and free amino acids. They are again acted upon by the dipeptidase and aminopeptidase and to get free amino acids. To get free amino acids. Now free amino acids, they are absorbed. Amino acids, they are absorbed from the small intestine. The three mode of absorption, first one is the sodium dependent symport. So it is a type of secondary active transport. Suppose this is intestinal mucosal cell. So this is sodium. Sodium absorbed inside the cell. Along with the sodium, there is absorption of amino acids. L amino acids, they are absorbed with the help of transporter, with the help of carrier protein. So this is known as secondary active transport. Here utilization of energy is indirect. So sodium will move outside, free sodium will move inside in exchange of potassium. So sodium will move outside, potassium will move inside with the help of sodium potassium ATPase. And here is utilization of energy. ATP is required for this process. So this is indirect use of energy. So this is known as sodium dependent transport or symport of the L amino acids. So this is the one mode of absorption, that is sodium dependent transport. Then another way is the various sodium independent transport systems are available. Independent transporters are available. Sodium independent transporters. So there are 5 types of sodium independent transporters. The first one is the transporter for neutral amino acids. Neutral amino acid that specifically involves tryptophan, phenylalanine, tyrosine, valine, leucine, isoleucine. So neutral amino transporter for neutral amino acid. Second, transporter for acidic amino acid. Acidic amino acid, that is aspartic acid, glutamic acid. Then transporter for basic amino acid. Basic amino acid, so that include arginine, lysine. Then transporter for amino acid, which is amino acid and glycine, that is proline and glycine. Transporter for proline and glycine. And next one is the transporter for beta alanine. So these are the transporter which are sodium independent. So these are the 5 sodium independent transporters which are available for the transport of amino acid into a intestinal cell and into a circulation. So 5 types, 1, 2, 3, 4, 5. Neutral amino acid, specifically tryptophan, also includes the transporter for phenylalanine, tyrosine, valine, isoleucine, leucine. This neutral amino acid, acidic, aspartate, glutamate. Then basic amino acid, arginine, lysine. Amino acid and glycine, they transport with the with the same transporters, and beta alanine transporter, sodium independent transporter system. Then the type third in the transport or absorption is the Mr. cycle. Mr. cycle. This is also known as gamma glutamyl cycle. There is involvement of glutathione, and this is for the transport of glutamate and cysteine. Transport of glutamate and cysteine. It is a costly cycle. There is requirement of 3 ATPs for the transport of single amino acid. It is a costly cycle. Involvement of glutathione, that is gamma glutamyl cysteanyl glycine. Glutamate and cysteine absorbed by this cycle, and 3 ATPs are required for the transport or absorption of single amino acid. So these are the 3 modes of transport or 3 modes of for the absorption of the amino acid. Okay, now coming to the disorders. Disorders in the digestion and absorption. Disorders. So we'll just enumerate the disorders. The first one is the Hartnupp's disease, which is very, very important. The Hartnupp's disease. There is defective absorption of tryptophan. There is defective absorption of tryptophan from the intestine. Mainly the tryptophan, other neutral amino acids are also affected, but tryptophan is the major concern. So tryptophan in our body is converted into niacin. When there is defective absorption of tryptophan, it affects the niacin, and it will manifest as pellagra, classical 3Ds, diarrhea, dementia, and dermatitis. These are the symptoms in the Hartnupp's disease. Now the second important disease is cystinuria, lysinuria. In cystinuria, lysinuria, there is defect in the absorption of cysteine, ornithine, lysine, arginine. Cysteine, ornithine, lysine, and arginine. There is defective intestinal as well as renal tubular transporter of cysteine, ornithine, lysine, and arginine in cystinuria or cystinuria, lysinuria. Third one is the iminoglycinuria. Iminoglycinuria. In this defect, there is excretion of proline, amino acid proline. There is excretion of glycine because of defective transporter. So iminoglycinuria means proline and glycine excretion because of defective absorption of proline and glycine. Then the fourth one is the pancreatitis. Inflammation of pancreas causes the malabsorption because it affects the pancreatic secretion, which is important for the protein digestion. So pancreatitis and cystic fibrosis, they are particularly involved in the malabsorption of the protein. And the fifth one is the celiac disease. In the celiac disease, it is also known as protein losing enteropathy. The person is hypersensitive to the gluten, which is specifically present in wheat, barley, oat, rye. Okay, so celiac disease, it is also known as protein losing enteropathy. There is loss of proteins from the intestine, and person is hypersensitive to the diet containing the gluten, which is component of wheat, barley, rye, and oat. So that is known as gluten sensitive enteropathy or protein losing enteropathy or celiac disease. So these are the few disorders which are related with the digestion and absorption of the proteins. Once again, we'll revise this. We discuss the two types of peptidases, that is endopeptidase. So peptidases are of two types, that is endo and exo. So endopeptidase, exopeptidase. Endopeptidase will act on the internal peptide bond, exopeptidase will act on the external peptide bond or terminal peptide linkages. They are hydrolases. These peptidases, these are hydrolases. So digestive enzymes belongs to the class hydrolases. The enzyme commission number will start from the 3. OTH, oxidoreductase transferase hydrolases. So these are hydrolases. So two types, endopeptidase, exopeptidase. The important endopeptidase, these are pepsin, trypsin, chymotrypsin, elastase. And important exopeptidase are the carboxypeptidase and aminopeptidase. Aminopeptidase. These are the important enzymes. They are involved in the digestion of protein. Absorption, sodium dependent, sodium independent, and Mr. cycles. So this is all about the protein digestion and absorption. Keep watching. Thank you.